Hi, and thank you for giving me the opportunity to present my work at OHBM 2023. My name is Alex. I work as a research engineer in the clinical and experimental neuropsychology lab at the University of Geneva. And in this presentation, I will be talking about COVID-19 and how you might actually have long COVID without being aware of it. And I have no disclosure in this presentation. So I won't go into too much details about the acute COVID-19 and the pandemics. I believe you are all well aware of it, but I will focus more on the long-term effects of the infection. So for some context, last year, the World Health Organization defined the post-COVID-19 condition as the continuation or development of symptoms 3 months after the infection, with these symptoms lasting for at least 2 months. And they reported that 10 to 20% of the infected population may develop such symptoms. Now, aside from this specific post-COVID-19 condition, around 60% of the population had at least one persistent symptom after 4 to 6 months, decreasing to 30% of the population after 1 year. So in Geneva, we built the COVID cohort as a collaboration between the university and the university hospital. And so the project aimed at investigating the long-term cognitive effect of the infection in 121 participants that have been tested positive for COVID-19 between March 2020 and February 2021. An exhaustive battery of neuropsychological and clinical assessments was administered 6 to 9 months after the infection, and a subset of 50 patients agreed to conduct an MRI with anatomical diffusion and resting state functional scans. Now, while the initial aim was to evaluate the long-term effect as a function of the severity of the acute respiratory symptoms, we were surprised during the clinical assessments of participants, because one of the first results was that some patients were not aware of their deficits, notably in the memory domain. And so last year, we raised the presence of anosognosia, which is the unawareness of deficits as part of the long-term sequelae of the infection. Now, to measure the self-awareness of symptoms, we computed self-appraisal discrepancy, or SAD scores, for 5 domains of cognition. And the measure was basically the difference between objective and self-evaluated performances. Now, looking at the chart, we see the objective deficits on the horizontal axis and the complaints on the vertical axis. And so it places the self-awareness of symptoms as a continuous score between the overinterpretation in the top left and the unawareness of symptoms in the bottom right. And so from this, we asked ourselves, can we differentiate between clinical phenotypes related to the self-awareness of symptoms? And the answer is yes. We used a data-driven approach with an agglomerative hierarchical clustering on the 5 SAD scores. And we chose here 3 clusters, which was the model with the highest stability. And so the results were one cluster of 20 patients here in orange that could be hypothesized as having higher awareness of symptoms, one cluster of 34 patients in green that could be hypothesized with lower awareness or anosognosia, and finally a third cluster in red of 59 patients with normal self-awareness. Now, before I proceed to the neuroimaging segment, let me quickly answer a question that you might have, which is, why the brainstem pons? And so models put the brainstem at the roots of consciousness, while being part of the ascending reticular activating system, or RAS. And then lesion in the brainstem usually induces coma. And it has been hypothesized that the pons might promote efficient interaction between subsequent cortical structures. Now, on the other side, the brainstem appears to be affected by the infection. A meta-analysis reported that 25% of the neurological events involve the brainstem. And then studies reported structural alteration of the white matter and functional alteration as observed through FDG PET scans. So with this in mind, we probed for association between the self-awareness of symptoms and brain functional dynamics of the pons. To do so, we computed co-activation patterns, or CAPs, using the pons as a seed. So the idea is to select frames with high pons activity and then feed the selected volumes to a k-means clustering algorithm. So this could be basically be seen as a decomposition of the seed-based connectivity into distinct but overlapping co-activation maps, or brain states. And in our case, the approach yielded high stability for 5 CAPs that are displayed here. Then we compared the temporal aspects of the CAPs between our 3 clusters. So the metrics were first the occurrences of CAPs, which is the proportion of selected frames expressing a particular CAP, and the average CAP duration in the number of time points. And so the results show that patients with lower awareness, or the cluster number 2, displayed lower occurrences of CAP2 and more occurrences of CAP5 than normal participants, or the cluster number 3. Then no difference could be observed in the group with higher awareness, cluster 1, and when analyzing average durations. But if we focus on the region involved in this difference of CAPs occurrences, we see that both CAPs showed co-activation in the cerebellum and co-deactivation in the orbitofrontal cortex. These structures are known to be affected by the infection. But then CAP5 was characterized by a focused co-activation in the thalamus and co-deactivation in the visual and somatosensory area. Finally, we discarded the clusters or phenotypes and considered the self-awareness as continuous data. Utilizing the partial least square, or PLS framework, we ran data-driven analysis to evaluate the multivariate correlation between behavioral and imaging data. And so the models yielded significant correlations between the SAD scores of fatigue and the occurrences of CAP 1, 2, and 5. In other words, and to simplify a little bit the chart here, it means that lower self-awareness of fatigue was associated with fewer occurrences of CAP 1, and especially CAP 2, as well as more occurrences of CAP 5. And so this kind of suggests that CAP 5 is a functional state that could be detrimental to the self-awareness of symptoms of fatigue in our case. So to conclude, we showed the presence of clinical phenotypes associated to the self-awareness of symptoms in the post-COVID-19. We propose that the self-awareness might be a better predictor of the long-term cognitive sequela than is the severity of the acute infection. We showed specific brain network dynamics of the pons in patients with lower awareness, such as this brain state CAP5 that appeared to be detrimental to the self-awareness of fatigue. And so we think that these alterations may have arisen from a disruption between sensory inputs and memory processes. Nevertheless, these are our interpretations, and the actual origin of this self-awareness disorder, as well as acute predictors, are not known. Briefly, there are evidence of similarities between the long-term effects of COVID-19 and either accelerated neurodegenerative processes or chronic fatigue syndromes, which are both coherent with our clusters. But on our side, we are still investigating. So that's it. I quickly wish to thank my colleagues at the University of Geneva, as well as the extended COVID-COG team, both at the University Hospital and EPFL. And I also wish to thank OHBM for putting up this exciting meeting, and thank you all for your attention. For more information, please come talk to me at the poster number 2447. And here are some relevant papers from our